This material is made available to you by or on behalf of the University of Melbourne under Section 113P of the Copyright Act 1968. It may be subject to copyright. For more information, visit the University Copyright website.
Saying when the presentations are on, and then also what groups you have to peer review as well. Any questions about that, or it makes sense? Yep. I tried to work it out so that out of the 5 presentation days, you need to come to 3 of them. So there's the day that you present and then 2 other days as well. And so it's all should be spaced out. So everybody should be also assessed by the same number of people as well. Okay. And the comment that I put down the bottom about what the, I've made up a Microsoft form so you can assess. I'll send out that link later, but they're the things that you will get assessed on. So there's questions about, or people will give you a rating out of 5. So 1 lowest, 5 highest for each of those different components, and then also your presentation style as well. So you know what other people will assess you on, and then you also know what you'll be assessing them on. So today is the last content lecture for the course. We're going to be going through tissue engineering. So tomorrow's one is going to be about the term paper. You'll then have presentations. And then in the final week, I'll go through a review lecture and what is in the final exam lecture as well, give you examples. Again, like with the tissue engineering lecture on Friday, there is more information in the 3rd edition of the book as compared to the 2nd. So I've only listed the 3rd edition book chapters, and like with all the other lectures that I've done, please read through this, and the stuff that I give you is additional information on top. So I know with the tutorial you've gone through tissue engineered skin, artificial skin, yep. I wanted to add more onto that, mainly because for about 6 years this was the research that I did, so I know a fair amount about it. And I wanted to add on the stuff that you've done in the tutorial and again take it towards a biomaterials focus. So there's a few tables. I'll go through a few tables. So this is a review paper that came out in 2011, and it listed a whole different range of commercial products that at that time were currently on the market. So in the tutorial you would have learned about ones that are just the epidermis, ones that are just the dermis, and then ones that combine both of them together. Yeah. So I have done this lecture in a few different iterations for a few years. Each year I always go and check online and I see how many of these companies still have products that were were listed as that in 2011, but then also ones that are still companies. A lot of them don't exist anymore. And when I search for them to find out about them, a lot of the information that you find is in more review papers. Because I think for the case study for the last on the skin, you have to talk about another product, is that correct? Or compare it to one and go through advantages and disadvantages? Did I read what you have to do correctly? Pardon? It's one of the options. So the ones that I've got on here, they're all examples that have been on the market, though some of them have been taken off the market, or they have in a way failed as a product, whether or not there was something wrong with it, or they don't make enough money, and the company wasn't viable, and some of the companies aren't around anymore. You'll often find with industry, companies change names, products change names, so I may not have been able to find it underneath that name, but it still maybe exists. So these are examples of commercially available epidermal constructs. So they just contain the epidermis. Take note of, particularly for these ones, the graft type, the cell source, and the biomaterial. So for all 3 of the tables, these are the things that I've got listed. And so think about in terms of what the product is, what the product is doing, how to make the product for all of these different things. So the first couple of ones are cell-based. So they don't have any biomaterials involved. Did you guys watch a video about Fiona Woods and the cell spray? Have you guys heard of Fiona Woods before that? And then I wanted to link that, and I won't show the video that I've usually shown because you've already seen one. If you're now linking this work and stuff, go back to what you learnt in regulations as well. So who can remember how the technology of cell spray works? So there is a patient, it's usually a burns victim or a burns patient that has a fair amount of their skin that has been exposed to burns. So the skin graft may be possible, may not be, depending on how much skin is left. They're taking a small sample of the patient's own skin, they chop it up using enzymes, they put it into a solution, and then you spray it back on. So think about regulations, getting something like this through the regulatory pathway, and how, in a way, this product has become quite successful and easy to regulate. So usually when you're developing tissue engineered strategies, you have to go through a lot of the TGA regulations because you're taking cells from a patient or from a source, you then take them to another lab, you then grow them up, all of the regulations that you need to get around that process. Whereas the very intelligent thing with the cell spray is you have a patient in theatre, you take a bit of their skin, within 30 minutes it's digested, and then you put it back onto them. So the bit of tissue doesn't leave the operating theatre. The operating theatre is all sterile, the kit that you get to do it is all sterile. So in terms of regulating a product and getting it to market, a lot of the steps that you usually have to go through with tissue engineered products, that has been eliminated because of how the process is. But with all of these, and I'm going to go through cell sheet technologies in a little bit, because that, while the device or the construct that goes onto a patient doesn't go with a biomaterial, they're using a biomaterial to help make it. All of these are autologous. So all of them, the cells have come from the patient. If you're growing up cells from a patient, there's always then time involved. And in some of the other ones, I will go through actually how long it takes. Do you go through in the tutorial how long it takes to grow a little tiny piece of skin? So I will go there. The other thing is that all of these are permanent. So they're being developed to go on the patient and stay on the patient. None of it actually gets removed. So looking at the materials that they're using, if you're using a material and you want it to be permanent, you usually, or particularly because it's for skin, are you going to want something that degrades away or something that stays in place? So the MySkin Celltran is actually, they're growing it on a surface. They're using that surface to transplant it onto the patient, and then they remove the bottom layer of the material off and the cells are left. So even though it's a cell-seeded scaffold, that bit of material is removed off. The other two, last two, so a recombinant protein. A recombinant type of protein is going to degrade over time, be reabsorbed. And then they also have a fibrin sealant. I would have mentioned fibrin with blood clots. So it's another type of protein that's in there. At some point, it will also degrade away. So in terms of the dermal constructs, there is a lot more available. You'll notice how with these, the majority of them are cell-free. So on Friday, when I went through, there's kind of two ways to do tissue engineering. You get the scaffold, you populate it with cells, and then you implant it, or you get the scaffold, implant it into place, and let the patient's own cells integrate. So with the dermis, Sometimes with these ones it will then depend on the thickness of tissue that you're trying to regrow or regenerate. If you have a patient's 3rd degree burn, usually the burns will go down to the bone, and if you have a look at how thick that skin is, in terms of growing a tissue, that is a substantial size that you need to do. So it's usually why it doesn't necessarily work to grow a tissue in the lab to then implant back. Sometimes it works better, depending on how big the wound is, to put a scaffold into the patient and then let the cells naturally infiltrate and regenerate the tissue. So if we have a look at, then also take notice of where the cells are coming from for the ones that do have a cell source. Neonatal, and again autologous. So either the patients, neonatal, so babies, so an adult cell, you can get stem cells from adults, and usually adult cells, they've already picked what phenotype they are, or they're harder to change. And by phenotype, I mean the type of cell and how it behaves. Embryonic, so if you were to take embryonic cells, which then there is a lot of ethics around, you're able to manipulate them or change them a little bit more. And then so the neonatal, it's not quite the same as embryonic, but you have more flexibility with what you can do with them compared to the adult. But neonatal skin, so it's usually foreskin from patients that have circumcision, so you then have an excess of skin. So skin that will usually go into medical waste is then a source of fibroblasts. So it's one way to use it. You'll also find anything that has cells that are not from the patient, and it's a product that you want to reproduce. There's then also quality control with the cells. You have quality control with the manufacture of materials. You're going to have quality control with manufacturing the cells. Can you get the cells to behave the same way each time? Primary cells that I would have mentioned in a few of the lectures ago, once you isolate it from patient and grow it up in culture, it will only behave like that for a certain amount of time as well. The neonatal cells will behave a little bit longer than adult cells. So it's one way to get a population of cells that will behave the way that you want them to behave, for also product quality control and having something that is reproducible. You'll also notice with these, not all of them are permanent. Some are semi-permanent. Some are temporary and you take them out. So are you putting something in place that is just stimulating the body to start to regenerate the tissue? A lot of these, until you actually look at the company and see what it is for, will be very different. So for skin tissue engineering, the amount of different types of uses in terms of burns compared to wounds, then depending on what type of wounds they are, usually dictates very much what type of products that you use for these. So the last one is the commercially available epidermal dermal constructs. So it's full thickness skin. You've got both layers. Comparing the amount of companies that have them for both dermal and epidermal, there is a very, very small amount. Appligraph still exists. I think there is actually a version of the other three that are still available. Whether or not I have been able to find them, whether or not they're still available under the same name as that, but there are still companies that have them. I usually then show this manufacturing video. They don't have it on their website anymore. so I can't show it. But what they do is they go through the steps to how to make these. And because this is what I used to do myself, I know how long it takes. So think about in terms of you're developing a product, I'll explain the steps to you, how long you think it's going to, well, I'll tell you how long, but I guess the costs involved with all of this. So you'll have banks of cells, you'll have your keratinocytes, and you'll have your fibroblasts. You usually grow your keratinocytes on a permeable membrane for around 7 days. Oh no, sorry, before you do that, you usually make a dermis. So the dermis is usually fibroblasts in a collagen gel. You'll grow that from anywhere from 1 day to a week. You'll then put keratinocytes on top of that. You let the keratinocytes grow for about 7 days. Then you bring the whole thing to the air-liquid interface. By bringing it to the air-liquid interface, that means that the top of the keratinocytes, just like your skin, are exposed to air. The other cells are still sitting within the media, so they can then get the nutrients. You then usually grow that construct for between 3 to 4 weeks. So it takes 3 to 4 weeks for it to mature. By taking it to the air-liquid interface, you get what's called stratification. So you get the different layers of the epidermis. Our top layer of skin is dead cells that just shed off. This is what happens when you bring it to the air-liquid interface. So you're getting something that looks like skin grown in the lab, but it takes roughly about a month to create. Like with cell culture, you usually have to feed it or change the media every 2 to 3 days. So the amount of man hours that are involved with that is a lot. If you think about the quality control that you need to go through with all of the different components with this, the cells, the media, all of the reagents that you use. That's why these sorts of products can cost a couple of $1,000s for a patient to have. You've got in vitro skin or skin grown in a lab that you're then putting onto a patient, can cost a lot of money. However, depending on what they're used for, usually these sorts of products, they're quite small, are generally limited to wounds and chronic wounds that can't heal. If you're thinking about a patient that has burns to 60 to 70 part of their skin, I guess product-wise, is it then, or how much do you think is going to cost? If a little piece of skin around that big costs a couple of $1,000s to have a patient who has burns, it's not economically viable. But for the patients who have the wounds and get these, even though it costs a couple of $1,000s, the success rate of them is quite high, depending on what type of wound you have, if it's the correct one for use. Yes. So while it does cost a lot of money, have any of you known anybody who has a chronic wound? Or seen chronic wounds? So I guess in terms of chronic wounds, these are wounds that usually don't heal. They're classed as a chronic wound if they have been open for more than 3 months or not healed within more than 3 months. But a lot of the time, they don't heal. So you have something that is open all the time. The pain involved with them, the constant changing of the dressings, all of this costs a lot of money. And depending on where the wound is and what type of one is, can usually impact on the patient's quality of life. So while it may sound like a lot for a couple of $1,000s to have skin that's grown in a lab for a therapy, quite often it is a good option because it has quite a high success rate. So I wanted to show you one more example. So this is this is from a company, their material is called Novosorb. It's a BTM. BTM, well, the acronym will go through in the video that I showed you. This is a company, who has heard of Polynovo? Polynovo is a company in Melbourne. So this is a company in Melbourne that makes these polymer scaffolds. According to their website, a lot of them are still in clinical trials at the moment, but they're developing synthetic materials for different skin tissue engineering applications, and one of them is a burns, for burns as well. So I'll show you the first one about what their product and what it does, and then I'll show you the second one about their back grafting and elasticity. And it's one of those things where you're growing a tissue in the lab and you wanting to have it the same function as what a patient does. If you've ever seen a patient with quite bad third-degree burns on their body, depending on how old they were when they got them, there's often a lot of scarring. With scarring comes tissue contraction, and sometimes patients that have them, they will then, particularly if it's around the shoulder area or elbows or any joints that are moving, if you have contraction, quite often their limbs can't move. So you have a patient who, while may have gotten over the third-degree burns, they are then have limbs that can't be moved. So one of the big things with developing therapies for burn victims is being able to develop something that creates skin like what the patient had before. And one of the big things with skin is elasticity, because it is one of the very big factors in the patient's quality of life. So even if the patient recovers from this and their skin is contracted, what do you think the rest of their life is going to be? And if they have contraction, you can then have surgeries to loosen it, but quite often if it's scarred and is contracted, going into surgeries to cut it, then you might be able to loosen a little bit, but then it often creates more scar tissue. So here is a video that goes through how these scaffolds, or the scaffold technology, so material-wise, and how they're using it to grow skin.
NovoSorb BTM, a biocompatible and biodegradable polyurethane matrix, is composed of 3 layers: a wound-facing 2mm open cell foam matrix, a polyurethane bonding layer, and the temporary sealing membrane to physiologically close the wound. The open cell foam creates a matrix of interconnected cells of varying sizes. This provides a structure for dermal cells to infiltrate, proliferate, and vascularize, as the NovoSorb foam is slowly resorbed and hydrolyzed over an 18-month period. NovoSorb BTM is applied to a fully debrided wound and trimmed to abut the wound edges without overlapping. BTM should be lightly tensioned to ensure apposition to the wound bed, and mattress staples may be inserted over wider areas. Staples are applied at regular intervals to secure in place. Initially, you will see hemoserous fluid uptake into the foam. Progressively, over the next 3 to 4 weeks, the matrix will fill with cells and vasculature. In the weeks following application, closer inspection at the cellular level shows the proliferation of new vasculature, as well as macrophages and a fibroblast producing extracellular matrix, forming a connective tissue scaffold that replaces the biodegrading BTM. Once the neodermis has fully formed, a simple Blanch test will indicate that the BTM is ready for delamination. The sealing membrane can be removed. The new dermis is ready for closure according to the clinical choice of the surgeon.
So they're using, it is a synthetic material that they want to break down within around 18 months, and I'll show you some histology slides as well. What it's breaking down into are more smaller, I will say biocompatible, because within this aspect or within the application it is. I want to say it breaks it down into polylactic acid. On the website it has information about it, of what you have a polymer, they're then taking into account what components it's breaking down into, and then you obviously, if you're wanting it to degrade, what is going to be left and what effect that has. And they've chosen a component that it's either easily secreted or absorbed into the body, but it shouldn't have as many toxic effects. So this is from a patient, should explain what the patient has gone through.
Okay, this is back graft on BTM. patient 5 for the burn trial. This was injury was sustained on October the 6th, which is just over 6 months ago. Obviously, the BTM would have been in situ for about 5 weeks before being grafted. And this is now the result then. It's sort of 5 months post-grafting. And that is as soft as you can hope for. Lovely result. With marked fading of the interstitial mesh pattern, especially on areas like here on the shoulder.
So I have seen a lot of videos and a lot of pictures of patients with burns and wounds. That patient's skin after that period of time, for how big the area of skin on his back, it's phenomenal. The elasticity, can you kind of still see, they may have done a little bit of skin grafting as well because of how big it was. Quite often with skin grafts you can see kind of a hatched, they put patient skin, you put it through a grater, which then stretches it out and puts it on, and you can see like the diamond shapes. So quite often you can tell if a patient has had a skin graft, that's what it looks like. So they may have had a little bit on there, because I guess also if you've got an area that big on your back, how are the cells from the outside? For the cells on the outside at the edges of the back to integrate through that tissue, it's going to take a while, so maybe putting a little bit of skin over the top, they may have done it. I didn't look into the details. But for that area of a patient to get elastic skin like that, it's pretty much unheard of. So I am very interested to see on the website, it said that they're still in clinical trials. I think some of them started around 2011, and I think maybe the other one was 2015 if I'm correct. But I did see a surgeon talk in Adelaide last year who is a burn surgeon and plastic surgeon and works with these materials a lot. And for a surgeon to say these materials are fantastic, and the outcome that they have for the patient and how similar the patient's tissue grows to behave and look like normal tissue, for a surgeon to say that that's not something that they've developed is pretty much fantastic. So I will be quite interested to see. But it is a company that they're making them, they don't have them on the market yet. But these are some histology slides of the scaffold that has been implanted. So if we're looking at what histology is, pink tissue, purple are cells. So this one is 6 months after it's been implanted. So you saw the scaffold it's a porous material that cells are infiltrating. So you might see all of the holes where it hasn't quite, the tissue hasn't integrated and infiltrated yet. And you still might see a lot of purple, which you do in the 6 months one, so there's a lot of cell infiltration. Look at also how big the area of the scaffold is. After 9 months, you have a lot of the tissue up the top, and there's a little bit still remaining, still quite purple, still a few little bit of holes, and it looks like those holes are more towards the centre of where the scaffold is. So how long does it take the cells to migrate in through the scaffold? After 12 months, tiny, and after 18 months, it's not there. You're also starting to see, and this is more of a side note, the organisation of the tissue, and this is something that I want to talk about at the end of the lecture in terms of tissue engineering and creating tissues. Here, to me it looks quite homogeneous in a way. It's not going to be because you're going to have different tissues, but it's a great big area of pink. If you looked zoomed in, you would see differences. Now compare it to this. You're starting to see different levels of organisation of the tissue, different structural features come out, which to me looks more like skin. So think about that and different organs when you're trying to create tissues, and that's what I want to talk about at the end for essentially why tissue engineering of tissues and organs is very complicated and hard to do. So I talked a lot about different cardiac devices, and because when I was doing the cardiac lecture, I gave an example of a tissue engineered blood vessel, I didn't want to talk more about it. I wanted to talk something else, or another tissue engineering really application or a device for cardiac patients is, so patients who have myocardial infarction, you have, something happens and you have death of tissue within the heart. So for patients that have that, are there then tissue engineering applications or technologies that can grow tissue to replace it? And the answer is yes. So this is where a lot of people are working on cell sheet engineering. So the figure on the top is and that technology is quite, when I say quite old, probably around the 2000s, people were publishing it, where they are taking a scaffold that is biodegradable, feeding cells onto it, growing that tissue, and then placing it on the patient's heart tissue to replace the tissue that has died, and then recreate live tissue. You have a biomaterial there. So the other thing particularly with cardiac patches is, what does the heart do? It pumps. And has anybody ever seen cardiac cells growing in cell culture and how they beat? So if we're wanting to create tissue that goes onto a heart because of a patient that has had an MI, you're wanting tissue that's able to function like heart tissue, that can beat. Sometimes if you have a biomaterial in place, does that then restrict how the cell material, how the tissue beats, contracts, releases? It could, depending on what it is. The other thing with cardiac tissues, as compared to some of them, other ones, cardiac tissue, they have quite close cell-cell junctions. So the cells are grown in culture very close to each other because the electroconductivity will run through the cells, and that's one of the ways that it's able to do it. So you're also wanting to culture something where the cells are in contact with each other. They can align by themselves, and if you have a material in place, particularly a porous one, can the cardiac cells then do that? Sometimes yes, sometimes no. If you've made a material or a scaffold that has alignment, you can get cells to grow all in one direction. And so that is where cell sheet technology comes in, and you can grow sheets of cells. If you're wanting to grow a thick tissue, like the example is on image B, you can then layer them one on top of each other and create a tissue. I think I mentioned before, you can only grow things so thick before the cells in the middle no longer get any nutrients without a blood supply. So that's the same for tissues in our body, but it's also the same for growing tissues in a lab. It's around 200 microns. So if you're growing something that's roughly around 400 to half a centimetre, so 500 microns to around half a centimetre, depending on how porous it is and how much media or nutrients can infiltrate through and how much waste can release out. If you have something that's quite dense and you don't get that diffusion of nutrients, you'll get a necrotic core inside. So by creating individual cell layers, putting them on top and then putting them into a patient is one way to do this. So this is where cell sheet engineering has come in, and it has been around for quite a while. So anybody who has grown cells on a 2D surface, you have cells, you put them on a 2D surface, they will grow until they're confluent. But what I mean by confluent is there is no more room for the cells to grow. A lot of cell types, when you grow them on 2D surfaces, they won't grow on top of each other. They'll grow until they meet the next one and then they'll stop. And there's only a certain limit to what they'll grow. To then take them off and either divide them up or do whatever experiment you're going to do with them, you have to put an enzyme in contact with them. It's called trypsin. The enzyme chops up all little bits around the edges of the cells so the cells can lift up. So if you're growing a tissue where you want these cell junctions to be nice and tight, you don't want there to be any disruption of the cell matrix that the cells have used, If you grow them on a 2D surface and you then have to put trypsin on them, it disrupts all that. So there are polymers that have a property called lower critical solution temperature, LCST. Depending on the properties of the polymer, you can tune where this LCT temperature hits. Essentially what the LCT does is at one temperature it behaves one way, at another temperature it behaves another way. And for the cell sheet engineering, at 37 degrees, the properties of it cells like to adhere to. You bring it down to about 20 degrees, it changes, the cells then don't like it, and the cells lift off. When we culture cells, we grow them in an incubator at 37 degrees. You take them outside of the lab and you work in a cell culture hood. Over time, it will drop down to room temperature, or depending on how cold the air conditioning in your lab is, to around the 20, 25 degrees, usually closer to 20. So you're creating biomaterials that then you're not using it on the patient, but you're using it to grow these sorts of cells. And because you're creating the cells, you'll see it, I've watched videos, people just grab both ends of the cell sheet with tweezers and you can lift it up. And it's just a layer of cells. So if there's the cardiac cells and you can see them beating, you can then lift them up and you're not breaking the cell-to-cell junction, so you're not disrupting what you've just grown. And cells that are making their own extracellular matrix, because you're not putting any enzymes in contact with them, you then have them, the natural ECM that these cells make is still there. So this is just an example of one where I found for corneal engineering. So the patient has an issue with their cornea, depending on what it was, whether or not it's diseased or they've done damage to it. They've taken cells from the patient's tooth, or not tooth, mouth, oral mucosa. They've grown it up on this LCT, on a surface that has, I think it's got around, 20 degrees, the LCT property. So 37 degrees, you're growing a nice comfortable wet cell layer. Take it down to 20, you can lift off this sheet, and they've transplanted it onto the patient's eye. So this is what the cell sheet looks like, and then they've put it on the patient. So you're able to grow the patient's own cells and place it on their cornea. The cornea, remember, is only a few layers thick, so you don't need to grow a great big tissue. So people are doing this for cardiac patches, for cornea, but for all different, I've even seen people grow small amounts of liver tissue as well, if you're wanting, and you can also grow these. People are starting to look at growing kind of mini organs in a dish to be able to look at different functional properties of an organ rather than, really carrying out animal studies, or there are things that you can do before you go into animal studies, a way to create a 3D tissue. So the last few slides that I want to touch on and show videos, and before, keep this in mind when we talk about the last slide. I think I've talked about decellularized tissues before. I've gone through some examples of 3D printing, growing tissues, thinking about the structure of something. How, I guess, realistic do you think it is to 3D print a whole organ? For me, I think it's something that, if it is obtainable, it's not going to be obtainable for a fair few number of years. But there are people who are taking whole organs from patients that couldn't necessarily be used for transplant, decellularizing them, and then repopulating them with patients' own cells and getting them to function as that normal organ. So this is just an example of different decellularization processes and how you take it from up in A, the native organ, and what you do to remove it. So the other thing with decellularizing tissues and how you, depending on how you do it, you can remove everything from the patient that is immunogenic. Or sorry, you can remove anything from the donor organ that is immunogenic before placing it into the recipient. When I also talked about some of the biological heart valves, particularly ones that come from their porcine derived, so animal derived. For a commercial product, they were fixing the tissues as well. So these sorts of organ transplants, it's not a fixed tissue. You're just removing the cells and then repopulating it. So it's going to have different properties and it will behave differently to a fixed animal tissue in some of the products.
Improve lives.
I won't go through all of this.
For more than a decade, biologists have been able to turn stem cells into beating heart cells in a petri dish. And bioengineers have had some luck getting stem cells to grow on scaffolds to make simple, hollow organs like tracheas and bladders. But just how hard would it be to make a large, complex organ, like a heart, from scratch? Getting beating clumps of cells to organize into a three-dimensional heart requires scaffolding. At the Massachusetts General Hospital in Boston, Harold Ott and his team start with the scaffold that nature provides. They're using a technique called decellularization to strip away all the living cells from a heart. The owner of this specimen died only a few days ago. It will sit in a specialized vessel for about a week, while chemicals wash away its DNA, lipids, soluble proteins, sugars, and all other cellular material.
Building, if you want to renovate it and repair it, you go in, you knock down drywall or get rid of cabinets and stuff like that. And basically we're going into the matrix and doing that by perfusing solutions and detergents. We're getting rid of all the cells.
Afterwards, all that's left is a pale, white matrix of collagen and other structural proteins that once held everything together. Stripping away the donor cells removes the antigens that can cause the body's immune system to reject the new organ. The scaffold can then be repopulated with healthy new cells that are immunologically matched to the patient. The team hopes to one day completely reconstitute working hearts from a scaffold like this. But today, they're focused on how they might develop parts of the heart using the same process. They've taken a valve and part of the aorta from a decellularized heart and sewn it onto a cadaveric heart.
This part here is the aortic root, including the aortic valve and the ascending aorta. And this has been decellularized, so this is a decellularized graft sewn onto a cadaveric human heart. You can see here the right coronary artery button and the left coronary artery button sewn onto that decellularized graft. Same thing here, this is the pulmonary artery and the right ventricular outflow tract here of the cadaveric heart. This is the graft, the decellularized graft, and then been sewn onto the heart.
Harold and his team want to check if their graft would be able to do what these structures do in real life, prevent fluids from flowing back into the heart and feed oxygen.
So I'm going to stop it here. You can watch the link in the video. I wanted to show you another one about a different organ.
They use a CT scan.
So it's by the same group.
If Dr. Frankenstein
And you can see what they're able to
alive
do.
today, he'd probably want to stop by the Massachusetts General Hospital in Boston. Here, scientists are harvesting dead organs and returning them to life. But instead of giant green monsters, these researchers are creating hearts, lungs, limbs, and kidneys that can one day be used to replace failing organs in people. Reporting in Nature Medicine, Harold Ott and his team describe their vision of the future and how they created a functional transplantable kidney in rats.
What we try to do here is to regenerate organs for transplantation, organs for patients with end-stage organ failure.
Using donor organs that would not normally be suitable for transplantation, Ott's team reanimates them by first washing away native cells with a soapy solution. What's left behind is a natural scaffold of structural proteins that can be repopulated with stem cells taken from a patient.
What's fascinating is that you're able to, with that process, preserve the blueprint of the organ, but it's completely washed empty. It's a mere a shadow of the original tissue. Now the major hurdle here was to get the cells, the viable building blocks, back into the right spots within that organ. That we did through creating literally a pressure gradient across the kidneys. So we were able to suck these cells into the tissue, into the kidneys, into the right spots.
The organs were placed into chambers that simulate the environment of a body, complete with oxygen and nutrients. After a few days of growth, something remarkable happens.
The tissue became functional, so these kidneys started to make a rudimentary urine. In the last step of this experiment, as the ultimate proof of principle, we were then able to take these kidneys from culture and transplant them in an animal model. So we took that final step to take the kidney out of the bioreactor and then treat it like a transplant organ, like a donor organ, and replace the native kidney in an animal with this engineered kidney.
But it's not always that easy.
We had to figure out how much vacuum can be applied to the kidney in order to provide just the right amount of tissue gradient to deliver the cells, but not to damage the matrix. Of course, if more vacuum is applied, we had quite a few kidneys blow up in a jar.
Hiccups aside, laboratory-grown organs appear to be closer and closer to fulfill their tremendous promise.
The reason why I'm most excited about this recent publication is because it shows the platform character of this technology. The technology can be applied to any tissue or any organ that can be perfused by its own vascular system. Ultimately, this is a very translational kind of research. It's very much focused on driving this all the way down to the bedside, getting this to the patient.
So I guess, to me, it is quite, my thoughts on that, it is quite interesting. They have an organ, so they have the scaffold there, but then it's the repopulating it with cells. All organs need a blood supply, and that is probably the hardest thing, or one of the biggest complications when it comes to tissue engineering and growing tissues and organs in the lab. So the other thing I wanted to talk about in terms of some of the limitations, well, not some of the things that we haven't overcome as a hurdle to be able to grow tissues and big sizable quantum tissues in the lab, or even organs. So I put 4 different types of tissues: skin, cartilage, blood vessel, and bone. So when I look at all of these, I see different structures within the individual tissues, and on quite a small, but it's a piece of skin has multiple layers. A piece of cartilage has multiple layers, and these multiple layers then have different properties. So if you take cartilage, for example, there's 3 cartilage layers before it starts to get into bone. These 3 cartilage layers, the collagen in each layer orientates a different way. The cell type in each layer are different. And the hard thing, and what we haven't been able to do, is design equipment or design materials or design technology that is able to print these multiple levels of structure in something in the lab. Theoretically, it seems quite easy to do. Practically, it's not. It is quite hard. Even the printing of the cartilage. I know many groups that are trying to print the different layers and grow it all together. It's then hard to get the layers to, in a way, integrate and behave the same. Particularly with cartilage, you need to put mechanical pressure to grow the tissue. The different zones or the different layers have different requirements in terms of what you want how you want to treat them to grow into cartilage. So it's very hard. And then the other thing is, like I said before, blood vessels. If if you have a sizable tissue or a sizable organ, how does it get the nutrients? It's through blood vessels. We don't know how to grow them fast enough yet. One of the biggest issues with biomaterials is the lack of vascularization, and the amount of people around in different labs trying to come up with different technologies is, there is too many to count. There is so many people trying to do it, but still it's something that we haven't achieved. And to me, they are probably the two big limitations of being able to grow something in the lab that is more than about half a centimetre thick, which is why I said growing a whole organ, I think we still have a long way to go. So even just from the materials point of view, to be able to develop a scaffold to create those sorts of structures is very hard, and we haven't been able to do it yet. Hopefully one day we can. But that's why I guess also the decellularized matrix, you have the scaffold there. So you have one of the components. You don't have the vascularization. You can feed vascular cells in there, and when you implant it into a patient, it's going to have its own cells going in there. But it's still, you have one of the limitations. The other thing with the decellularized tissues is that I didn't state was they all come from patients, or they all come from people. So like with organ transplants, you're always then reliant on someone else, something to happen to them before you're actually able to get the material that you want to grow the organ. So I think in terms of a tissue engineering field, there is still a long, long way to go, and there is still a lot of research being done about it, particularly from both industry and academia side as well. And I only did touch upon it a little bit, but there is an elective that you can do on cell and tissue engineering. So that would go into a lot more detail about all of this. So that is the last slide that I have. So does anybody have any questions? It usually does go patients with skin, whether or not it's a burn that they're trying to get healed better or with wounds, the hyperbaric oxygen therapy increases wound healing. So,
but have they tried it with the biomaterial?
As in to grow it?
No, as in like with stable nerves or something like that. Have they experimented with that in terms of vascularization?
They may have. I haven't looked into what they were doing in the clinical trial and whether or not, I know for some things they usually do, it's one of the control groups. You have a patient that just has it, a patient that's undergoing the hyperbaric, and then with and without the different biomaterials. They do often look at it because it is something that is just used in wound without putting in that. But I wouldn't be surprised, but it would depend on what the patient was being treated for to whether or not. Yeah. Okay, well, I guess, thank you very much. I will see you guys tomorrow. If you have any questions about your term paper, if you want them answered, bring them tomorrow. If not, I'll see you guys later.